Welcome to the Keystone Biology Review on genetics. We have to start by talking about Gregor Mendel, who is considered to be the father of modern genetics due to his pea plant experiment. Why is it important that he studied peas? Well, there are several reasons. First is that peas, like these other types of angiosperms, plants that have flowers, are able to go through a process called self-fertilization because they produce both pollen, which are male gametes, as well as eggs, which are female gametes. So they can self-fertilize, they can also be cross-fertilized. So it's very easy to control the mating. Other reasons would be that the traits that he was able to observe were very distinct and that there wasn't a lot of subjectivity to those traits that gave him some great quantitative data. As well, for plants, it doesn't take a lot of space to grow them. Their reproductive cycles are very short and they can produce lots of offspring, so more data to use for analysis. Prior to Gregor Mendel, he actually wanted to study how to create the best hybrid. Ultimately, though, he was able to come up with some laws of inheritance. Prior to Mendel, it was hypothesized that parents had distinct traits and that they were able to put those traits together to pass on a blend to their offspring. So if we had one parent that was tall, one parent that was short, their resulting offspring then would be that blend of the two, and that all of the offspring that you would get from this medium plant would only and ever be medium. But what Mendel saw in his pea plants was that if he crossed a tall and a short, he only got tall. There was no medium. And really interesting, after self-fertilization, he not only had tall, but he had the reappearance of this recessive trait. So he was able to figure out that this would be recessive and that it could be hidden by a dominant trait based upon his experiment, and that there was no blending, that these, we now know them as alleles, were able to stay intact and be passed from parent to offspring. So the principles that he was able to help establish were the law of segregation. And without even knowing what DNA was, he was able to state that the two alleles for each gene are going to be placed into different gametes. So meaning that if a parent had a tall and a short allele, that each one of those egg or sperm cells that they made only ever had either a tall or a short allele for that trait, not both, that they were able to segregate or separate from one another. He also was able to hypothesize the law of independent assortment. In this one, it states that the inheritance of one gene, let's say eye color, doesn't affect the inheritance of another gene, let's say hair color, and that each of those genes can be inherited independently. You don't always have to inherit brown hair, brown eyes, but you could inherit brown eyes, blonde hair, that each of those two different alleles does not influence the inheritance of the other. And then lastly, law of dominance. When you have two different alleles present, one is going to be dominant and will be expressed. And the only way to express the recessive allele is to only have recessive alleles. So what are alleles? Alleles, for every gene that you have two of the same alleles. Or you can have two different alleles. So you can either be homozygous, having the same alleles, or you could be heterozygous, having two different alleles. And alleles are going to be different versions of the same gene. So blue eyes, brown eyes. The recessive allele is only going to produce a phenotype if there is no dominant allele present. So therefore, you need to have two copies of the recessive allele in order for an individual to show that recessive allele. Phenotype refers to the physical trait. So in this case, it would be purple. Genotype refers to the alleles that you have for the trait. So in this case, they would be homozygous, dominant for the genotype, two capital letters would represent two dominant alleles. Heterozygous genotype would be one of each, and homozygous recessive would be both recessive alleles. Besides complete dominance, where even if you have one of the dominant alleles, but you also have a recessive allele, you show the dominant trait, there are what we call non-Mendelian traits. For instance, if a trait follows codominance, if you are a hybrid or if you have a heterozygous genotype, then you are going to show both of those traits. So both, in this case, blue and yellow fur. For incomplete dominance non-Mendelian traits, again, if we look at the hybrid, the blue allele cannot hide or mask the yellow allele, and instead with incomplete dominance, you end up with a blend in the phenotype. So blue blended with yellow would make green. So this is similar to the blending hypothesis, but what we know today is that these are actually alleles, and the expression of those alleles would just blend together in incomplete dominance. You can still separate these alleles, and you could end up with offspring with yellow or blue based upon the genotype of the offspring. Another kind of non-Mendelian trait are sex-linked traits. These are going to be traits that are found on the X or the Y chromosome, and therefore the inheritance pattern is going to be different between males and females. For X-linked traits, the males tend to be ones that express more of the recessive X-linked traits than females, because if they have one X, that they only have one chance of inheriting the recessive allele compared to females who have two X chromosomes and therefore have a higher percentage of chance that they're also going to inherit a dominant allele. Polygenic traits are also a non-Mendelian example, and this is because for most of our traits, they are going to be controlled by more than one gene. It's not as simple as one gene, one protein, one trait, but many of our traits actually follow more of a norm curve distribution and a wide range of phenotypes. Skin color is a great example of this. There are more than one gene that actually code for the production of melanin, and depending on how many of the dominant dark melanin alleles you inherit from your parents, you could have two parents that result in a wide range of phenotypes for their offspring. Multiple allele is another non-Mendelian trait, and just as the name suggests, it means having multiple alleles or more than two for a trait in a population. A great example of this is blood typing, where we don't just have an A and a B allele, and by the way, both A and B are codominant to one another. So you can see this here. If you have both A and B alleles, you have both the A receptor and the B receptor, but there's also an O. And in this case, O is recessive. So the only way to have type O blood is if you only have O alleles. So how can organisms create new chromosomes? Because diversity is of utmost importance for life. One of the ways that we can do this is through crossing over. So again, in my last video, I talked about these, which are your homologous chromosomes, and they are going to exchange pieces of their chromatids, so pieces of their arms, with one another. And now if you see, you have 4 chromosomes that are now genetically different from one another. They're going to contain a different combination of alleles because of crossing over. Other things that can happen would be random chromosomal mutations. So there could be a deletion where you remove a segment of the chromosome, a duplication where you have copied too much DNA and therefore have a longer chromosome, an inversion where you're going to flip the order of genes on the chromosome, and this can interfere with how you actually express those genes. translocation, taking genes from one chromosome and crossing them over with another, but these are not homologous chromosomes, so they're not going to code for the same traits. And now because they're in different positions, again, that can interfere with the way that you express these genes. Non-disjunction is also another chromosomal mutation that would change the combination of chromosomes that an individual inherits. So non-disjunction, just like it says, non, it doesn't, and disjunction, it doesn't separate. So in this case, you're going to end up with gamete cells that either have more chromosomes or less chromosomes than they're supposed to. And of course, this is going to interfere in the genome of an individual. And for genetics, you have to make sure that you know what the central dogma of molecular biology is. And this is because all living things are going to follow the same order of information. DNA that we all inherit then codes for RNA, which is very much like a disposable copy of that genetic code, which is then translated into proteins. And proteins are how we express our genes. So all life also uses the same types of RNA to make proteins. We use the rRNA, which is the ribosome, or part of the ribosome, that is going to be the protein manufacturer. We use the mRNA, which is that disposable copy of DNA that's going to ensure the correct order of amino acids. And we use the tRNA. The tRNA brings the amino acids and matches them up with the corresponding mRNA in order for these amino acids to link together to make a protein. So protein synthesis is going to happen in two main steps. You are going to have transcription and then translation. Transcription. Think here, if you were to transcribe something, even what I'm saying right now, you're probably going to keep it in English. So transcription is going to keep it in the same language. So it's going to take DNA and then it's going to copy it down into RNA. But translation now is taking RNA and now translating it into a different language, a protein language. So here you use RNA polymerase, to make messenger RNA. That's going to come to the ribosome, where the ribosome, rRNA and tRNA, is going to bring with it amino acids to make those proteins. The entire life that we have on our planet also reads their genetic code using the same codons. And we can note which amino acid is coded for from these codons using either a codon chart or a codon wheel. For example, if we start with DNA, DNA first has to transcribe itself into RNA. So we're going to use base pairing rules for this, but we're also going to remember that with RNA, there is no T or thymine. So instead, where we would put a T, we're going to put a U for uracil. So if this is my DNA strand, my RNA strand is going to look like this. T goes to A goes to U, C goes to G, G goes to C. And I'm going to complete that strand. Next, I need to translate messenger RNA into the protein. So I'm going to translate this codon, this 3-letter base, into an amino acid. I'm going to use the wheel to start. So here we have A, and then U, and then G. So this is methionine, which is going to be my first and my start codon. All polypeptides or all proteins are going to start with the same start codon, AUG. My next one, ACG, let's use this. So we have A, and then C, it's in this box, and then G, ACG, so that's going to be THR, my amino acid next. And then last we have UAA. So let's do this. U, A, it's somewhere in this box. A, it's my stop. Stop is not an amino acid, but that's going to be my stop codon. So I'm going to end up with something that looks like this. So what's important to note is that the genetic code is redundant. Look here how there are a number of codons that are going to code for the same amino acid. The genetic code is unambiguous, meaning that the same 3 letters for a codon in us is going to always produce pro, just like it will in bacteria. There's no denying what amino acid is going to be coded for, and that it's punctuated. So all amino acids are going to start with the start codon and end with one of these 3 stop codons, just like you're writing a sentence right now. You start with a capital letter and end with punctuation. And once those proteins are made, then the ribosomes are going to send them on a pathway to release them either into the cell or out of the cell onto the membrane. So if a ribosome makes a protein and it's not attached to the ER, that protein is going to stay within the cell. But if the ribosome makes a protein and it's attached to the rough ER, that protein is going to travel from the rough ER through vesicles to the Golgi, where it can be further modified, and then to the cell membrane, or the protein could just go directly to the cell membrane. And last, what happens if the genetic code gets altered in some way? So besides chromosomal mutations, there are also point gene mutations that are going to happen at a smaller scale level. So a point gene mutation is changing one base on the DNA sequence. And so there's lots of different examples of point mutations. If there is a point mutation where it's a substitution, where you're just swapping one base out for another, the different ways that it can result in a mutation could be a silent mutation. So even though we change amino acids and change the codon, it still is going to code for the same amino acid. So therefore, there should be no difference to the protein structure. You could have a missense mutation, and this one is going to code for a different amino acid than the original, and this might interfere with the protein structure. And there is a nonsense mutation, in which instead of coding for an amino acid, it codes for a stop codon, and that can prevent the protein from even being formed in the first place. Besides substitution, you can also have frame shift mutations, where you add or remove a nucleotide, and this is going to change every single codon after that mutation. So some examples here as to what would happen. If you have a substitution mutation, these tend not to be quite as severe, although they can be. So if the original sequence was the fat cat ate the rat, notice here the hat cat ate the rat. So a little bit different meaning, but relatively you would be able to make sense of that. And therefore the protein shape might not be too dramatically altered. But if we have a frame shift, if we delete, we're going to shift everything over. So now the act, ada, tet, her, et, it does not make sense, and therefore it's significantly going to change the protein. Same thing with insertion. It shifts the reading frame. So again, it makes it very hard to read. The f, tat, ert, er, t. Because of this, it's going to greatly change the protein structure. So what does that mean if I change the protein structure? Well, here's an example. If you are changing the expression of a protein, this one is actually a neat protein. It's called the sonic hedgehog protein that's going to deal with placement of fingers and toes. So if there is a protein change, then that's going to lead to a phenotype change. And in this case, instead of having the normal 5 fingers, it now can result in extra toes on a cat paw if a gene is suddenly turned on in both areas on the inner and outer paw. And so what we can now do with our understanding about how genetics and DNA works is we can then impact society with genetics. So we can create things like genetically modified organisms that maybe are going to allow for better pesticide resistance and less of the harmful chemicals that we would even have to consume with our food. It could allow for us to modify a bacteria cell so that it can produce something like insulin, and that we can create these transgenic organisms so that we can use them for pharmaceutical industry. Without this, you would have to extract insulin from something like a pig pancreas. And not only is that the not exact same type as this would be for humans, but it is timely and cost ineffective. We can use what we know about DNA and something called restriction enzymes to be able to take DNA from a crime scene and figure out who the criminal suspect could be based on similar DNA bands and DNA fingerprinting. And we can also use what we know to create personalized genetics to make sure that we are prescribing the correct medication for the correct disorder. So at the genetic level, we can see how effective each one of these medications could be. And we can potentially even cure diseases through things like gene therapy, where we're going to use a CRISPR-Cas9 system to genetically alter individuals or cells of individuals that might be affected with a debilitating and devastating illness. I hope this helps. Have a great day.